This is section 4.2, talking about cellular structures. So the first slide is talking about the cell membrane, also can be called the plasma membrane. The job of this is to separate the inside of the cell from the outside of the cell. And we call it semi-permeable, which means that it's only going to allow certain things in and out of a cell. And we do that by having these proteins that are embedded in the membrane, and those proteins get to determine what gets to come in or also what gets to leave the cell. And we call this a lipid bilayer. So the whole cell membrane is two layers basically of really tightly packed phospholipids, kind of one on top of the other, and they arrange themselves in such a way that things really can't get through. So this is what a phospholipid looks like, and it's also what makes up that bilayer. So we have a polar head, which is made of mainly phosphorus, and then we have these two nonpolar fatty acid tails. So because the head is polar, it's going to situate towards the outside of the cell, or towards the inside of the cell. We call it hydrophilic, which means water-loving. So since most of the outside of the cell and most of the inside is water-based, that's why they kind of situate like that. And then the tail is hydrophobic. It doesn't like the water, so the tails are going to face each other. So if you look at this picture on the bottom here, here's the polar heads in blue facing either to the inside or outside of the cell, and then the pink hydrophobic tails kind of face each other. So we make kind of this fatty middle part, and then the polar phosphate kind of outer part. What we call the fluid mosaic model is basically just describing how a cell membrane behaves. So in 1972, this was the most widely accepted model of how cell membranes act. And basically, it's just saying that they behave more like a fluid rather than like a solid. So cells are able to kind of change shape. They're also embedded with all those proteins, giving it that mosaic pattern. We have all different kinds of things embedded in there, including cholesterol, carbohydrates, plus the proteins. So it's able to kind of move with the body and also change its shape to fit certain areas of the body. The next cytoplasmic organelle is called cytoplasm, and it's a gel-like material in the cell that holds all the organelles in place. It's mainly consisting of cytosol, which is just a watery substance. It's about 80 to 90% water. It's not going to contain any organelles, but it is going to help us with biochemical reactions. So this is where most of metabolism is going to take place, where we're going to dissolve things that the cell has been eating. Next is the cytoskeleton, and just like it sounds, cytocell, and it's the skeleton of the cell. So it's the scaffolding that basically crisscrosses across the cytoplasm and helps maintain the shape and integrity of the cell. It's also going to kind of help anchor the organelles in place, and sometimes it allows us to squish the cell without actually crushing the organelles, so maintaining that integrity of the actual cell. Two things make up the cytoskeleton. We have these things called microtubules and microfilaments. And first one, just like it sounds, they're very small little tubes. They're kind of thick, but yet they're hollow little cylinders, and that's kind of shown, those blue tubes up here in the picture. And then the microfilaments are these other structures kind of crisscrossing throughout that cytoplasm. So they're a little bit longer, a little bit thinner, and again, kind of helping keep the shape of the cell. For external structures of a cell, we have the flagella, which is a long, thin structure coming off of the cell membrane. That's the picture to the right here. Basically, two things that can help a cell move or kind of tumble forward to catch food or to move to another part of the body. There is only one human cell that has a flagella, and that's the human sperm. Otherwise, out in nature, we would find a few more like one-celled organisms that might have a flagella. So, and it's not like a wagging tail, it's more like a boat propeller movement. So it kind of spins and tumbles, really can't steer where it's going. So it's kind of a guessing game. The bottom one is cilia. And cilia are just little hair-like extensions coming off the cell membrane. They're made from microtubules, and they do more of a sweeping motion. So you would find these like in the lungs or the bronchi area of the human body. They help sweep the mucus up from the lungs to the back of your throat, where you kind of swallow it down all day long with your saliva. So a nice rhythmic sweeping motion for the cilia. Next up is the nucleus. The nucleus is a membrane-enclosed organelle found in the middle of the cell, and it has to be a eukaryotic cell. Having a nucleus makes it eukaryotic. This is usually the largest organelle in the cell, except in cases of plants. Usually the vacuole will be the largest, followed by the nucleus. And this is where most of the cell's genetic information is going to be stored. We store it on these things called chromosomes. They're little X-shaped structures. And it contains all your genes, which code for all the proteins that your body makes and runs everything your body does. The nuclear envelope protects the nucleus. It's a double membrane, so it's 2 layers of phospholipids. Sorry, 2 layers of lipid bilayers, so you're actually getting 4 separate layers of the phospholipids. And it helps separate the nucleus from the surrounding cytoplasm. So we have kind of an inner and an outer membrane. The outer membrane is going to link on to the rough ER, which you see pictured down here, so that we can take something that's made in the nucleus, like a ribosome, carry it all the way out and through the ER, and it doesn't really even have to touch the cytoplasm. And how things get in and out of here is through nuclear pores. It's just a little tiny opening in the nucleus. Again, they are going to regulate what's coming out and what's going in. Very few things get to actually come and leave the nucleus. Right in the center of the nucleus is the nucleolus. This is where all ribosomes are made. And ribosomes are where we're going to make protein. So we make the ribosomes and then we kind of ship them out into the cytoplasm. And then it's kind of out in the cytoplasm where we end up making the protein. And that's a whole separate process that we'll actually learn in the next chapter. Next organelle is called mitochondria. And mitochondria are also membrane-enclosed organelles, mainly in the eukaryotic cells again. And these guys are considered your power plants of the cell because they're actually using organic compounds, so like the foods that you eat, and converting it into a usable energy source called ATP. ATP stands for adenosine triphosphate, and it is the cell's basic energy source. So we use ATP to move all of our muscles, to help divide our cells, to basically run and do everything the body needs to do. So if you look at this mitochondria here, here's the inner membrane. It's kind of folded so we get a little more surface area. Outer membrane is going to be a little bit smoother. And something also kind of unique about mitochondria, they do actually contain their own DNA. So, and that's on this next slide here. So they're able to actually reproduce asexually. So they can kind of, whenever we need more energy, we can make more mitochondria and increase the amount that our body is making. So the number of mitochondria per cell really depends on how much energy that cell needs. So muscle cells are going to have upwards to hundreds of mitochondria, whereas, you know, something like a skin cell, which doesn't have to use a lot of energy, is probably going to have significantly less than that. Next up is the ER, also known as the endoplasmic reticulum. This is a whole kind of connected series of phospholipid membranes. And there's a couple different functions for the ER. First, it's a transport system, so it helps move protein that is coming or the ribosomes too, that are coming out of the nucleus, kind of helps move it from one side to the other, so it doesn't really have to touch the cytoplasm. And then there's also a synthesis portion, and that's where we actually make proteins, because there's ribosomes on the outside of rough ER. So, and on smooth ER, we're also making lipids. So we're doing a couple different making or synthesizing of a couple different compounds. So on the rough ER, or abbreviation RER, this is studded with ribosomes, giving it that rough appearance. That's why we call it that. And the ribosomes, again, make protein, and then they're transported to the Golgi apparatus via something called a vesicle. And it's just a little membrane-bound sac, again, protecting the protein so it's not touching the cytoplasm, not getting dissolved. And then the smooth ER, it's called smooth because there's no ribosomes attached to it. And this is the one that makes the lipids. And we can use those lipids to kind of rebuild our cell membrane if we need to, or rebuild any of the vesicle sacs, or all kinds of stuff that are made of lipids in the cells. So it's also going to help us store some calcium, and it's also going to help us break down some drugs. So some of our organs, like the liver, who does a lot with breaking down drugs, has quite a few smooth ER in those particular cells. And they're all connected. So smooth ER and rough ER do touch one another, and it's all an interconnected membrane. The smooth ER and rough ER transport their stuff to the Golgi apparatus via a vesicle, and the Golgi apparatus is like your UPS station. It's going to modify, sort, and package those substances, get them out of the cell, take them kind of to wherever they're needed to go then in the body. So they also help transport lipids. So again, like I said, rebuilding some of the membranes found in some of these other organelles. And you do find the Golgi in both plants and animals. A vesicle is just a small little compartment. It's made of a lipid bilayer. There's a whole bunch of these attached to the Golgi and the ER, and that's again how we transport things kind of in and around the cell. So the inside of that vesicle may be a little bit chemically different from the surrounding cytosol, and that's just because we don't want whatever's in that vesicle dissolving before we're ready to use it. So we classify vesicles by what's in them and also what they're doing. So if it's a transport vesicle, just like it sounds, it's moving it from one location to the next. We also have these vesicles called lysosomes. Those are formed by the Golgi apparatus, and they're filled with some pretty strong enzymes that are going to help break down waste materials, other cell debris. So they're kind of like the garbage disposal of the cell, getting rid of all the waste that you would find in a cell. And then peroxisomes are filled with basically a peroxide type chemical, and instead of getting rid of the waste, they now break down a little bit more of like the organic type substances, so they are going to help with metabolism a little bit. They also do a little bit with toxic substances, so again you find them in the liver and the kidney, where we're trying to break down maybe some medications that are being taken by the human body, some other things like that. Alright, next up is a vacuole. This is found only in plants, and it's a membrane-bound organelle that has several different functions, but the main one being storage. So its main job is to store excess water for the plant, and it helps maintain what we call turgor pressure. And turgor pressure is what keeps plants standing upright against that force of gravity. You can often see if a plant is starting to wilt. It's probably because their vacuole is not filled with water. That's kind of why it's starting to fall over and not look its best. Other vacuoles in plants can also store pigments or toxins. So this is also what can give some flowers their color. And it's also what may make a plant taste bad when an animal tries to eat it. The cell wall is also only found in plants. It's kind of a rigid outer layer around the cell membrane. So you'll have the cell membrane first and then the cell wall. And it's there for protection and support. So again, there's going to be some pores. It'll allow a few things in and out, but it's mainly, cellulose, which is a very strong sugar, some proteins, and basically it's making sure that the plant, again, can stand upright against the force of gravity and also stay nice and protected from foreign invaders. Chloroplasts are also only found in plants, and these are the organelles that we use for photosynthesis. So human cells obviously don't have a chloroplast. We don't undergo photosynthesis. And we're going to learn all about this process in coming chapters. But basically, they capture the energy from the sun. They use that with water, basically, and they convert it into a sugar. And that sugar can be used then later for them to make their own energy. So instead of having to eat something, they just use energy from the sun, and they do it just one extra step than humans do and animals do. So all right, so you have a little section in your notes where you're going to practice naming some of these organelles. The picture in your notes is probably better than the picture here on the slide, but we have a few organelles listed there. I'll give you a second to try and answer this question. So question number 1 says, which structures in this cell are also found in prokaryotic cells? So if you remember from the first section, prokaryotic means they do not have a nucleus. So the only thing that they would have then that would be similar would be the cytoplasm and the cell membrane. So the answer is B. Question number 2, which features of plant cells are missing from this animal cell? And the structures that are found only in plants are A, cell wall and chloroplast. Animal cells do not have those. Question 3, what is the function of the structure labeled A? So A is mitochondria. So go back in your notes, check what mitochondria does. They're the powerhouse of the cell, so therefore they help make our energy, which is the ATP. All right, and then you have this section as well, so go ahead and try and fill this in. So see if you can give these a shot and figure out which organelle belongs to which description. And that is the end of section 4.2.